Good evening, everyone. Am I audible to you all? Good evening to all. Please confirm in chat if I am audible. Okay, so we will start with our first question. This is from Unit 1, that is the structure and function of biomolecules. Today we will look, basically it's biochemistry. Today we will be looking at the different biomolecules, which is your amino acid, proteins, nucleic acids, protein conformation, Ramachandran plot, a little bit of bonding. Okay, so some things which you are expected to know if you're writing CSIR NET this time. Okay, the first question comes from Ramachandran plot. This says that the following are four statements on peptide conformation, and you have to say the combination of the correct statements. Okay, the first one says glycine has largest area of conformationally allowed space in the Ramachandran plot. Second one is, yes, very good evening, Satyam. So we have just started. This is the first question on your display. A 20 residue peptide that is acetylated at N terminus and amidated at C terminus. They have phi and chi residues, phi and chi value for all residues in negative. As you can see, minus 60 plus minus 5. It means that they are both negative. It can be concluded that conformation of the peptide is helix turn strand. This is a motif. Helix means it will come like this, then it will take a turn, and then it will form a strand. This is what is helix turn strand. The allowed values for phi chi for amino acids in a protein, they are not valid for short peptides. And a peptide in which you have acetyl, then 4 amino acids, and then amidation. So basically, A1 to A4, there are 4 amino acids. They will adopt a well-defined beta turn, in which the dihedral angle of A2 and A3 will determine what type of beta turn it's going to be. So, anyone who would like to answer, which would be the correct answer? Anyone of you? Okay. So, Satyam, if you are saying A means, do you mean option 1? Okay, you're partially right, because glycine has the largest area of conformationally allowed space. This is correct. So A will be correct statement, because glycine amino acid is a type of flexible amino acid. It has a very small side chain. We know amino acids have carboxyl group, amino group, hydrogen, and R group, which is a side chain. The R group in hydrogen in glycine is just hydrogen. That is why it is a very flexible amino acid and it can bend in many angles. That's why it is said to have largest area of conformationally allowed space in the Ramachandran plot of phi and psi. Okay. Option B, so this is correct, but option B it says, first of all, any peptide, if it has to be resembling the dynamics of folding in the cellular environment, it should be having the capping. So, acetylation, amidation, these are nothing but these are capping on the protein, on the two sides of protein, which will be done for, they will be done for protecting the protein and to help it fold in a better way. So remember, the N terminus is always acetylated and C terminal is always added with an amino group or amidated. They will have phi and psi values of both negative, minus 60 degree and minus 30 degree for all the residues. It can be concluded that conformation of the peptide is helix, turn, and strand. So helix is, as I mentioned, it's a motif, but in Ramachandran plot, they have a conformation. They have a, if you look at the standard Ramachandran plot, then helix will be having the phi psi values in the lower left quadrant. Lower left quadrant means both the phi psi values, they will be minus. Even if you look at the chart, these are the torsion angles or dihedral angles for different secondary structures in proteins. You will see that the right-handed alpha helix, generally L amino acids are have more tendency to form the right-handed helices always. In this phi and psi, both are negative in value. Okay. Then if you see beta strand, in the question it was mentioned helix, then turn, then strand. Now if you take any strand, whether it is anti-parallel or parallel, you will see that while the phi is negative value, the psi should be positive value. But in the question it was mentioned that all the phi psi values, they are having negative value. Then how come in the structure we can conclude that the strand is present? If strand is present means you should also have the phi psi values which will be having a positive psi. Okay, that's why the statement is incorrect. If it had negative phi psi for all the residue, that means positive psi is not present at all, then it can be concluded that conformation is entirely helix. Turn and strand is not present. If turn and strand would have been present, you would have seen phi psi values according to that. That's why the statement is wrong. Okay. Now the third statement, allowed value of phi psi for amino acids are not valid for short peptide. When you take a particular peptide plane, this is a peptide plane, and you can see the two peptide planes are connected by C alpha. So C alpha, if you take CC alpha, then this particular bond, C alpha with the next peptide plane and C alpha with the previous peptide plane. Anyways, this is an entire amino acid. The central carbon alpha has two peptide bonds on two ends. This is one peptide bond and this is one peptide bond. But at least it is connected with a carboxyl group and amino group with a bond. Okay. And this bond being single, it can rotate. So that's why these dihedral angles are called as phi and psi. Okay. So when you have this, even a single peptide bond will have at least one phi and one psi. And they can be plotted on the Ramachandran plot. But it is saying it is not valid for short peptide. That's why it is wrong. Okay. And a peptide, wherever you see a peptide drawn like this, you just see what is in between the acetyl and amido group. You ignore these things. Okay. So A1 to A4 are amino acid. They adopt a well-defined beta turn, and the dihedral angles of A2 and A3 will determine what type of beta turn they are going to form. Okay. So if you see a beta turn, their types are generally based on the dihedral angles only, and especially the type 1 and type 2 is based on the dihedral angles of the second and the third amino acid. Because you will see their orientation is what is different in the two types of turns. Okay, so the correct answer over here is A and D. Option third is right answer. Hope you got that, Satyam, and all others. Okay, next is from carbohydrates. Which one of the following sugars will not reduce tolerance reagent? This is a small part B question, And sometimes such structure based questions can be given from carbohydrates, especially from epimers also, or even the disaccharide contents, invert sugar, and sometimes this kind of reactions, famous reactions. So anyone wants to guess the answer? Okay. Anyone? See here, the options 1, 2, and 3, if you see, we know disaccharides are connected by glycosidic linkage. Either you write the sugar molecule in the regular plane like this, or you write draw it as it is present normally. Anyways, this also has this four, five corners, and this one oxygen. Okay, so these are like hexo sugars only. And from here we know the anomeric carbon is there, which will be connected with the next carbohydrate. Okay, it's just that the plane is little different here. So these two, what you have, option 1, 2, and 3, these are disaccharides with 1,4 glycosidic linkage. See, whenever we will be drawing the glycosidic linkage, you just have to see beside the oxygen. So this is the one beside oxygen. The OH group is present below or the OH group is present up. Okay, if it is on the same side as the fifth carbon, sixth carbon CH2OH, okay, so if it is on the same side, then it will be a beta sugar. But if it is on the opposite side, then it will be alpha sugar. So if O is on this side, that means whatever bond will be formed, this will be alpha glycosidic linkage. In all these cases, you have alpha glycosidic linkages. The hemiacetal of the second sugar ring is not involved in the glycosidic bond. Hence, the reducing end is free. Okay, and will reduce the tolerance reagent. So this is what is in option 4. If you see the second sugar ring, um, you have to start from the second one, right? So this you have to start from the second one. That is, the sugar is flipped. Okay. The other bonds, they are with the sugars which will be having the alpha 1,4 glycosidic linkage. 1,4 means from the anomeric carbon to the fourth carbon of the another sugar. But in this case, in 4, you are having this anomeric carbon, and another sugar is also bonded with anomeric carbon. So it is flipped like this, unlike this one. Okay, so that's why this also is anomeric carbon. Both the anomeric carbons are involved. That is, it is 1,1 glycosidic linkage. In this case, it's a non reducing sugar. Whenever the anomeric carbon, in this case, the anomeric carbon is free. So, whenever you will have anomeric carbon free, one of them is bonded with glycosidic bond, but at least one of the sugar, the anomeric carbon is free. In that case, they will be able to to be capable of doing a reducing agent, to serve as a reducing agent. But here, they will not be a reducing agent. Yes, that's right. Very good, Satyam. That's right. Sucrose is a non-reducing sugar because it lacks a free aldehyde group necessary to react toluene's reagent and produce a silver color. Very good. So when you reduce something, the anomeric carbon reduces something, it becomes a acetyl group. But before, when it is just free, it is a hemiacetyl group. And hemiacetyl group means it will be able to reduce. The hemiacetyl group of both rings are used up in option 4. That is why they are not free to make the reduction. Hence, there is no free hemiacetyl group and tolerance reagent will not be reduced. That's why answer is option 4. Okay. Then question number 3, it is again based on a protein. The following observations are made on a 30 residue polypeptide. That unordered structure is observed in water, but helical conformation is observed in medium of low dielectric constant. So what exactly is low dielectric constant? It means that they are hydrophobic or polar in nature. It is a measure of polarity, you can say. If the dielectric constant is high, then you will not have, then it will be a polar environment. And if you have a medium of non-polar environment, you are seeing that in water is a polar environment. You are seeing that the peptide is taking an unstructured, unordered structure in a polar environment. But when you are exposing the polypeptide in non-polar environment or hydrophobic environment, it is taking a helical conformation. These are your observations, and based on these observations, you have to conclude which out of this is correct. Second observation is peptide is resistant to degradation by protease. We know proteases will cleave the peptide bond between amino acids in a protein, and if the enzymes or proteases are not able to cleave the protein, it means the protein is resistant to degradation. Probably there is a change in the peptide bond alignment because of which the enzyme is not able to recognize the proteins anymore. Red blood cells are lysed by the peptide. So, the red blood cells, if the peptide interacts with them, generally our cells or organs, they will not be, um, means they won't be destroyed or disturbed by the own cells. But this peptide will lyse the red blood cells. Beta mercaptoethanol, which generally has an effect on normal protein structure. So this is a reducing agent. Okay, beta mercaptoethanol is a reducing agent. It will reduce the disulfide bonds which are used to fold the proteins. So let's say this is a disulfide bond. Here is a disulfide bond. So they will reduce the protein. Okay, but how reduce means gaining of protons. Okay, so they will give the protons over here, and the disulfide linkages will get reduced. So the molecule which is making them become reduced, it will be reducing agent. That means they will provide the electrons. By getting the electrons, the protein will open up. Their SH groups will be reduced by gaining of electron from the reducing agent. But this peptide, it has no effect by beta mercaptoethanol. So which out of this can you conclude? Any guesses, students? And I hope you know that amino acids generally are in the form of L amino acids. That is what you mostly find in nature. So the proteins which I am talking about, generalized ones, those are present in the form of L amino acids. Okay. So one hint is that if they are showing some kind of abnormality, it means they might be a D amino acid. Okay, yes. What will be the correct answer, Satyam? Any guesses? Anyone else also can try if you want. So one is that peptide is D amino acid and is amphipathic. Amphipathic means they can interact with polar and non-polar both. Okay. Another one is it's composed of L amino acid and it is not. Peptide is composed entirely of L amino acid, and peptide is rich in disulfide bonds between D cysteines. Yes. Okay. You are saying that it is rich in disulfide bonds. Okay. So here you can see if you take D cysteines, then the disulfide bond formed with them will not be reduced by beta mercaptoethanol. That's what you're saying, Satyam. Because even if you take D cysteines, if the rest of the amino acids in proteins are in L form, but the cysteines are in D, but still they will be involved in disulfide linkage, right? And if they have disulfide linkage, then beta mercaptoethanol will still be able to break them. Okay, so it will have an effect on peptide, which means that the protein should not be having this disulfide bonds. Definitely not. Otherwise, if there is disulfide bond which can form between D amino acids also or L amino acid also, that will not matter. But if it had disulfide bond, then definitely beta mercaptoethanol will be able to break those. But if it is not able to break, means definitely this is not the answer. Okay. So the correct answer will be D amino acid. As I said, obviously it is not L amino acid. Otherwise, it would have been resistant to proteases, which are generally they can cleave the D. Generally, they can cleave the L amino acid protein. But here they are not. They are not responding to degradation. Means they must be D amino acid. Okay, so these two can't be correct, and disulfide linkage can't be correct because beta mercaptoethanol has no effect. That is why option 1 is the only correct answer. Okay. So generally in our normal proteins, what do we see? You would have heard about a protein folding in polar environment, that is in presence of water. All the non-polar groups will be exposed outside. Sorry, all the polar groups will be exposed outside so that they can interact with the water, and all the non-polar groups will be clustered inside. Okay, where they will be having hydrophobic interaction with each other. They will not be able to interact with water, so they cluster with each other. This is called as hydrophobic aggregation. Okay, and that's why the protein will be having a stability in presence of water. Probably helical conformation or strand conformation or any protein fold can be formed. But here they are saying it is other way around. In water, the protein is showing unordered structure. Okay, which means that it should be amphipathic. Okay, is it clear? Yeah. Okay, fine, Satyam. So yeah, I've given the explanation also. All right. So I'd like to mention about the Lakshya batch in case you are feeling that the questions we are discussing here is it's a little difficult, you're not able to understand. Then let me give you a reality truth check that this is the type of questions that are asked in exam. Okay, and if you're looking to clear the exam, you know CSIR is a very recognized and reputed exam, all India reputed exam, if you have to get into quality research in any of the recognized institutes all over India. And that's why this is the minimum requirement that they will ask so that you can get fellowship. They will generally ask scholarship funded students. If you clear CSR JRF, then you will be getting the scholarship. You have to clear interview and you can get admission in those institutes and start your research, and along with that you will get a good amount of scholarship every month for your other expenditures. So take up this exam seriously. Okay, in case you haven't been even haven't prepared before in a proper way, then this is your chance. Unlimited classes will be there till 6 months. You can talk about the duration till when you want the coaching, because you also have for gate 2026 the next year. Already this gate will happen in February. If you want it to clear next year, and CSIR also another time it will happen in this year, probably in June, July, August, or September. And you will get unlimited classes where all the units will be covered. 24/7 chat support will be there. A dedicated study portal where all the recorded videos, PPTs, everything will be uploaded. Study books, ebooks, questions for practice, and also you will get the Conceptica series free access, which are the revision tools, helpful, very helpful in case of exam. Okay, and the test series as well. So test series are the ones which will be AIMNET test series or any weekly, monthly mock test series. And along with that, this is a limited offer that we are giving. You will get free molecular biology internship as well. This is a hands-on training as well after the internship. So you can contact us on our toll-free number if you want to be a part of it. Okay, I think you should not miss this opportunity and definitely should avail it. Talk to our team. You can also mail at info@biotecnika.org. Okay, next question. How many hydrogen bonds involving the backbone CO and NH can be observed in an alpha helix? Alpha helix is a type of protein structure. How many such bonds consisting of 15 amino acid residues? How many hydrogen bonds will stabilize this structure? Anyone? You can take a guess. How are we going to solve this question? Okay. So see, the key over here is you have to just this a numerical type of question, you can say a hydrogen bond in alpha helix is from the CO group of the ith amino acid to the NH group of I plus fourth amino acid. Okay, that is every fifth residue you will find a hydrogen bonding. This is an alpha helix. Okay, so let's say this is our first amino acid, which is called as the ith residue or nth residue. We don't know if this is the first amino acid of alpha helix. Okay, so that's why we cannot give it a number such as 1, 2, 3, 4 or something. That's why we write it ith, then I plus 1, I plus 2, I plus 3, like that we will write. And always a peptide is written from N to C terminal. Whenever we take CO of ith residue, this is the ith residue. For this ith residue, our CO group, actually this is not the C alpha, this is the C alpha. For this C alpha, if it is the ith amino acid, the CO group will be present towards this side and the NH will be present towards the other side. Okay. So this is what you have, the CO of the ith residue, then the NH of the I plus fourth residue. This is the I plus fourth residue. Okay. You can see C alpha is there. Again, individually you have to consider N to C. Hence, the NH group will be present towards this side. So CO of I to NH, they will form a bond. Then the next bond will be just addition of 1. That's why it will become CO of I plus 1 to NH of I plus 4. Sorry, I plus 5. Okay. That means you have to start from here. That is I plus 1 CO group and I plus 5, that is the NH group. NH to CO group. Okay. So when you have a continuous 15 residue peptide, let's say these are the 15 residue. 15 residue means you can also write as I plus 14. You will need, see for this first 4 CO groups, anyways their bondings will start from the very beginning. But if these 4 NH groups have to bind, if this NH group has to bind, it will need 4 amino acids before it. If this has to bind, it would need 4, but it has just 1. If this needs, it needs 4, but it has just 2. Okay. So that is why the first 4 amino groups, even this one also here, they will not be involved in hydrogen bonding. Only the CO groups will be involved in hydrogen bonding. From the fifth amino acid, yes, they are connected with hydrogen bonding, but first 4 will not have. Similarly, the last 4 CO will also be not involved in hydrogen bonding. Okay. Because you just think about it, if the 12th amino acid, that is I plus 11, 12th amino acid, if it has to form a hydrogen bond, then it will require 12 plus 4. It is always I plus 4, no? So that means it would require the 16th amino acid. But 16th is not there, this entire protein is still 15 residues. So since 16th amino acid is not there, okay, that's why the last 4 CO groups will not form hydrogen bond. Okay. In between them, that is I plus 4 to I plus 11, these will be not I plus 11, I plus 10, that is the 11th amino acid. They will have hydrogen bonding from both their NH group and their CO group. Okay. So they will have complete 1 hydrogen bond. So total 7 hydrogen bonds in the center. And the last 4 will have half hydrogen bonds. Okay. Last 4. And the first 4 will also have half hydrogen bonds. So half means 2 plus 7 plus 2. So total 11. This is very easy to calculate. Whatever number you are getting, let's say you are getting a 23 residue alpha helix. If they ask you to find out how many hydrogen bond is missing, just subtract 4 from it. So 19 hydrogen bonds will be required to stabilize that particular helix. It's very simple. Hope it's clear to you, everyone. Okay. All right. Now, next question is based on some protein structure. Unit 1 is very similar with, where 1 has lot of resemblance with Unit 13 techniques. Because Unit 13 techniques are based on biomolecules and protein studies, whether finding the sequence or whether studying the structural properties, or separation of proteins, all these techniques It's very important to learn unit 1 for that. Okay. A circular dichroism spectra in the far UV region informs on the kind and content of secondary structures in a protein. So CD spectra, it is a very popular method to study what type of secondary structures are observed in protein. Secondary structure means the either the repetitive ones, alpha helix, beta sheet, or even the non-repetitive and random ones which are turn, coil, loop, etc. Okay, and near UV and tryptophan emission spectra, they will mention about the tertiary structure. Okay, shown in this are A, B, and C. These are intrinsic fluorescence emission spectra of X. This is showing the far UV spectra of X, and C is showing near UV CD spectra of X. Okay, so what you expect to know from here is what is the pattern of the structure that we generally get. Okay, and in each one of these techniques, this is far UV spectra, this is near UV, this is emission spectra. For each one of this, there are three curves which are shown. The black curve is at a condition, it is of protein X only, but at a condition where pH is 7. The green is at a condition where pH is lesser, 3, acidic environment, and red is pH 7 but in the presence of guanidine hydrochloride. So guanidine hydrochloride, it is actually a denaturing agent. It is called as a chaotropic agent, which can denature the protein. Now you would know 6 molar is a good amount, even if you take pH 7, but it would probably denature the protein. pH also, we know pH, if it is disturbed, the protein conformation might also be disturbed, because pH changes the hydrogen and concentration, and hydrogen and concentration can change the charges and the charges on the protein, and also the polarity on the proteins. Okay, hence the curves that represent the spectra, one is normal, another one is denatured, another one is also denatured because of chaotropic agent. So you have to identify the result from this experiment. Okay, that whether protein is fully folded at pH 7, or it is secondary structures are reduced at pH, or there is extensive denaturation both at pH 3 and pH 7, or there is just a change in fluorescence and near UV which indicate increase in hydrodynamic radius. So obviously the answer is option 1, because when you compare with the black line, that is protein at pH 7, the green line shows a clear change in near UV spectra. If you see, the black line is here, the green line and the red line, they show a clear change, a complete change, which indicates that at pH 3 there must have been a change in the tertiary structure. On the other hand, if you check the far UV CD spectra, it doesn't show much change. Is the green and black lines, they are not showing much change. Means that indicating that at pH 3, which means that there is no secondary structures are not disrupted. That is, no extensive denaturation, no change in the kind and content of secondary structure, which is possible in molten globule state. So we can see in far UV spectra there is a change in the red line, which is in guanidinium chloride, hydrochloride. And in guanidinium hydrochloride, it is possible that you will have a molten globule state achieved from the protein. So what is molten globule? When a protein folds, it will not fold entirely at once. So let's say this is a polypeptide chain. Initially, there will be just a hydrophobic collapse, okay, where some nonpolar groups are aggregated towards the center, and then some specific parts of the protein, they will be arranged. Okay, so this is how the protein folds. So when you are treating the guanidinium hydrochloride over here, it is possible that the helices or secondary structures, they are not disturbed, they are still there, but somewhat the protein loses its intricate folds and contacts. So somewhat the secondary structures are maintained. That is why in the far UV CD you can see that the green line and red line are more or less similar, which suggests that they must have been in the molten globule state. Okay, but red line protein with guanidinium hydrochloride, which acts as a denaturant, shows a further increase in the emission maximum. Black is okay, green is little bit at the higher level, higher intensity, maximum intensity, and red is even further. So that is only possible when the protein is little bit opened up, okay, which indicates that the tryptophan residues must have been further exposed. See, emission spectra, it is because of the aromatic residues which will um, absorb some wavelength of light and they will emit it back. Out of all the aromatic residues, tryptophan absorbs the maximum. So if you have the red color means it has been denatured a lot, okay. So that's why the tryptophan has been exposed as a result of treatment with denaturant, which also confirms with far CD spectra, where the red line is flat, indicating complete loss of even the secondary structure along with the tertiary structure. So green line, some extent the secondary structures are still retained, okay, but not in near UV, because near UV spectra will reflect a lot on the intricate secondary structures. So overall, sorry, far UV will show the intricate secondary structures. Near UV is generalized loss of structure. So that is why red and green line are both showing loss of structure. But when you compare with this, you can see that the green line is only loss of secondary structure, red line is completely loss of the tertiary structures as well, which is also confirmed from the emission spectra. The hydrodynamic radius for native folded protein and is this much, and for highly denatured state is 2.21. So you can see 4.75 and 2.21 for denatured state. That means the hydrodynamic radius, it decreases. What does exactly it mean? See, when a protein is properly folded, it occupies a little bit more area. Okay, it is in a arranged form, ordered form, so it will occupy a certain area. But when the protein loses its shape, it gets denatured, it will not only lose the surface area, it will actually become more denser, it will become compact. So basically, the radius will decrease, which is lesser. Hence, with denaturation, you can expect the RH radius to get decreased. That is the hydrodynamic radius, you can expect it to be decreased. But since statement 3 says it is getting increased, that's why statement 3 is also wrong. So option 1 is the right answer. Protein is fully folded at pH 7, because of which it shows a proper far UV, near UV spectra, it is showing a proper one. And acid induced molten globule at pH 3, where secondary structures are still intact and unfolded in 6 molar guanidine hydrochloride, where complete loss of 3D structure is taking place. Okay. Next is this question again on proteins only, but more than proteins, this question depends on amino acids. So, what exactly this question is asking? A lot of blah blah is given in the initial area, but you can ignore that. You can just see this is a wild type protein, and monoclonal antibody is generated against the wild type. We know that antibodies will be complementary to their proteins. That is why here you have a thick band of the thick band which you are getting in the blotting paper out of the SDS page and Western blotting result. So you are getting a thick band which shows that they are complementary to each other. But from this wild type protein, some mutations were done, and those were single amino acid mutants. Four single amino acid mutations were done. Okay. So they are monoclonal, all our monoclonal antibodies are generated against the wild type. Okay. But when you have when you have this protein B, okay, you saw that they had the same band. That means that monoclonal antibody which was raised against the wild type, it was also used to test how much they are able to bind with the B, how much they are able to bind with A, how much C and D, which are all mutants of the wild type. while in protein, while in the protein B, you see that there is almost similar band as the wild type, which means that if this protein was complementary, if this antibody was complementary to wild type, and it has similar amount of band in this under B also, which means that this antibody is also complementary, similarly complementary like the wild type, which means that between B and wild type, there is not much difference in the structure, even after mutation. So that is only possible when the single amino acid mutation is done such a way that the amino acid in that wild type and the changed one in B, they belong to the same category, or they have the same properties. Either both are polar, both are non-polar, both are aliphatic, both are aromatic. So some kind of similarity will be there the in the amino acid which has been mutated. Okay. In A, you will see little less band is there as compared to wild type, which means that in A, some mutation has been done, which is a little bit different from that um, amino acid of interest in wild type. So that's why not much recognition is there, little bit recognition is there. That means the property is slightly different. But in C and D, you don't see any band at all, which means there has been a drastic mutation. That amino acid, that single amino acid which has been changed, must be very, very different from the one of wild type. They must be having opposite contrasting properties. That is why it did not give rise to a proper protein. Okay. I mean, it could not be like the original protein because the conformation might have got changed due to the property. Okay. So we know these many amino acids are used. So let's talk about this B, which is very similar. So B, here you are given B. They are saying B is due to valine to alanine and B is due to G glycine to proline. We all know glycine to proline. Glycine and proline are completely contrasting amino acid. Glycine is a very flexible amino acid while proline is very rigid. So clearly 2 cannot be answered because if there was glycine to proline, then the structure difference would have been a lot. But since here we see the same similar band, that is why this cannot be the answer. Okay. And yeah, this is possible that valine to alanine is possible because valine gives rise to valine and alanine, they are both non-polar amino acids. Now, C can be due to glycine and proline, that is correct, because these are completely contrasting. And we can see also in the C there is nothing like that. Nothing, no bands, which means they should be contrasting. Okay. Let's see if D is correct. D is saying, I mean, option 3 is correct. D is due to, where again you don't see any band, isoleucine to leucine. Again, this cannot be correct because isoleucine, leucine are both branched aliphatic amino acid. But in D, you don't see any band, means the changed amino acids should be very different from the original one. So if original one is I and changed one is L, there is not much difference between them, which should not have changed the structure such that there is no band. So 3 also cannot be correct. And even C cannot be correct because C is saying valine to alanine. As we saw, you need a change. So option 1 is the right answer. Yes, is everyone following? I hope the explanations are clear to you. Shall we go to our next question then? Please confirm. Just wanted to remind the people who have joined late that our lecture batch is just starting. You can join this batch and clear the upcoming CSIR and GATE exams. Okay, next question is based on the denaturation of the Anfinsen's experiment, what he had done along with the lysozyme. In classical Anfinsen's protein folding experiment, the enzymatically active ribonuclease is treated with beta-mercaptoethanol and 8 molar urea. So following which the preparation was dialyzed to remove the beta-mercaptoethanol. First, see, beta-mercaptoethanol you already know now, it's a reducing agent. And urea is also a denaturing agent. It's a chaotropic agent, just like guanidine hydrochloride, which we saw in the previous question. So both of them were removed they were dialyzed so that they can remove the denaturant. And then the sample was, I mean, this is one option. Second option was sample was completely oxidized. So if the sample is oxidized, we know that the protein is folded with the disulfide linkages, right? So this is the oxidized form. But this sample, after being denatured, it was oxidized in 8 molar urea after dialysis. Trace amount of beta mercaptoethanol was added to the dialyzed sample, means where beta mercaptoethanol and 8 molar urea have been removed, and 8 molar urea was added to the dialyzed sample. So after dialysis, again 8 molar urea was added. So clearly, this cannot be the answer, because if you are dialyzing to remove the denaturing agents, then again if you add denaturing agent, then what is the effect? The protein won't gain its full enzymatic activity. They are asking which of the following steps will lead to regain the full enzymatic activity of ribonuclease. And Finson has had used beta mercaptoethanol and 8 molar urea to denature the protein so that its activity can be lost. And out of these steps, if you remove the denaturing agents, then it might gain the activity. But again, if you treat it with 8 molar urea, then it will again lose the activity. So this cannot make sense. Sample was completely oxidized in 8 molar urea. So this is also more or less same only. You are again oxidizing it, means basically you are promoting formation of disulfide bonds once again once the protein is denatured. But urea is not the one um, where the disulfide linkages will be encouraged to oxidize. For that, you need trace amounts of beta mercaptoethanol. You can't give huge amounts. You just need few little amount of beta mercaptoethanol, so that once the denaturing agents are removed from the protein, you should know that ribonucleases have 8 cysteine residues and they can form total 4 disulfide linkages. And these 4 disulfide linkages, they should be in the correct form. That means if the protein has to be catalytically active, they should be binding with the exact disulfide linkage, okay, with the right cysteine residue. For example, the 40th and 95th should bind, 84th and 26th should bind, 65th and 72 cysteine residue should bind. So what happens when you treat with beta mercaptoethanol and urea? You can see all the disulfide linkage are opened up, because urea denatures and beta mercaptoethanol reduces ribonuclease A, which results in the unfolding and complete loss of activity. Okay. Now from here, if you remove the urea and you keep the urea, but you remove the beta mercaptoethanol, then quickly the disulfide bonds try to form. Okay, because right, reducing agent is not there. So they will again try to oxidize. That is what is the second option. Okay, complete oxidation with only 8 molar urea without beta mercaptoethanol. But because of that, wrong disulfide linkages will be formed. If you remove all the beta mercaptoethanol, wrong and random disulfide linkage will form, and 8 molar urea will not allow the protein to become properly folded. Okay, but when you remove the 8 molar urea and keep some amount of beta mercaptoethanol, so the protein will try to come back in the original form, forming the disulfide bonds. But even if some wrong disulfide bonds are formed, they might have a tendency to correct themselves. Okay, so from here you will have slow oxidation where these will join, and you can also remove the 8 molar urea from here. So removing urea first and allowing protein to refold before disulfide bonds were restored with oxidizing agent, and it led to recovery of 90% of activity. Both urea and 2-beta mercaptoethanol removed by dialysis, ribonuclease A refolded spontaneously and they regained biological activity. In the form of stress response, this is a question on protein folding. The previous one was from protein denaturation and this is from protein folding. Bacteria synthesize a group of proteins called stress proteins or heat shock proteins such as DNA-K, DNA-J, GROL, GROS, and GRPE. Here, DNA-K is an ATP binding protein. I hope you already know that they have their eukaryotic analog also. These are in bacteria, but in eukaryotes, these proteins which help in folding, they are HSPs. Okay, you have HSP 70, HSP 60, like that. So DNA-K is a ATP binding protein which attaches to the newly synthesized polypeptide in conjunction with DNA-J. Which one of the following statements correctly states a step in the subsequent process of protein folding? So one is that the affinity of DNA-K to the polypeptide increases upon hydrolysis of the ATP to ADP. Okay. First, you have to see the diagram for this one. In fact, this is only the correct answer. See, the DNA-K is what has binding sites of ATP. Okay, when ATP is bonded, the DNA-K doesn't bind to the protein that well. Only when ATP is hydrolyzed to ADP, that time only the DNA-K is able to bind to the unfolded protein in a proper way, which induces conformational change and it might encourage the protein to fold in a proper manner. Okay, but this ADP has to be replaced with the ATP so that the DNA-K can now dissociate from the folded protein, because they can't remain associated with the folded protein, right? So for dissociating, it has to be again replaced with the ATP. In this process, ADP is lost and ATP is added. This is done by a nucleotide exchange factor protein, which in this case in bacteria it is called as GRPE. Okay. After the ATP binding, the protein is released. It may be in the partially folded form and it might go in that case to another protein, which is a chaperone system. It's a complex of chaperones where it might have the possibility of folding properly. Okay. So it's saying that DNA-J is an exchange factor that replaces ADP with ATP in DNA-K. No, that's not the case. Actually, GRPE is your exchange factor. DNA-J is just required to recognize your protein, and DNA-J, once it is bonded with the protein, it will recruit DNA-K. ATP hydrolysis is required for phosphorylation of DNA-J. No, ATP hydrolysis is required for more affinity of DNA-K with the protein. ATP hydrolysis is required for phosphorylation of GRPE? No. GRP is required for removing the ADP and replacing with the ATP in DNA-K. So option 1 is the right answer, that the affinity of DNA-K to the polypeptide increases when the ATP is hydrolyzed to ADP. That is when it is able to associate with the protein even better. Okay. Next question is from nucleic acids. So this is from September 2022. The figure below represents the denaturation renaturation profile of a double-stranded DNA in citrate buffer. Okay, you can see that earlier when the protein was in the double-stranded form, that is, it was not denatured. It was showing absorbance to be 1. This is the absorbance which it is showing. Okay. After that, it was denatured, the temperature was increased, and around 80 degrees centigrade, 80 to 90 degrees centigrade, you saw after 90 complete denaturation has occurred. That means all the double stranded DNA has opened up. Okay, and at that point, the absorbance was found to be 1.40. This is the relative absorbance, mind you. Okay, now the protein was cooled down, the temperature was brought down again to 30 degrees centigrade. But this time, even if it is brought down to 30 degrees centigrade, you could see that the protein could only bind, not protein, the double stranded DNA had absorbance only till 1.12 or something. Okay, we can say 1.12. That means all of the DNA did not get denatured. Okay, how much of the DNA did not get denatured? This much. Because if all the DNA would have got denatured, it would have got absorbance 1. So even if you have taken down the temperature, this much of the DNA did not denature. It did not renature. It did not come back as double strand. This much came back as double strand. Okay. So all you have to find is the percentage, because 100% DNA denature means it is 1.40. Okay. And so that's why you have to find out 1.12 means how much is denatured. You just have to say that the percent of DNA that remains denatured at 30 degree after cooling down from 100 degree. Okay. So what we have to take is, see, we cannot take directly 1.12. What we have to take is the difference between them, that is 1.12 minus 1. So this part we need. Directly with this we no need. How much is the difference? You want to know this much. Okay. So absorbance of DNA still in denatured form, which did not renature at all. This much portion is still denatured. Okay, so that is 1.2 minus 1, which is 0.12. Okay, so according to that calculation, because this one is 1, no, so that's why. So according to that calculation, is 1.40 is 100%. Okay, then if you take unitary method, 1 will be 100 by 1.40, and if you take 0.12, then it will be 8.57. That means less than 10% or less than 20% of the DNA will still be denatured. Rest all would have renatured if you cool down the temperature. Okay. So solubility of NaCl is greater in water than ethanol. What physical property of the solvent governs this difference? See, NaCl is a ionic molecule. Okay, ionic substance. So the solubility is greater in water, which is a polar substance, than ethanol. So polarity of ethanol is not that much. So what physical property is present because of which this difference occurs? Obviously, it's up upon the polarity. Okay, ethanol, water is more polar, and like dissolves like. NaCl also has charges on it. Water molecule also has partial charges on it. So the charges will help them dissolve properly. So we can say dielectric constant is what is the necessary one. Okay. The explanation is given. Okay. Then I wanted to discuss one more question. Yeah, this one. This is from a recent exam, last exam, latest exam. The surface electrostatic potential map of an 18 kilodalton protein is shown below. Okay, so these are some shadings of blue and red which is given on the surface, and they denote positively and negatively charged surfaces. All right, we can see that more of the surfaces which are exposed outside, they are blue in color, which means they have positive charge, and red is present but less amount is present. Okay, so which out of these proteins would be more likely to bind to this enzyme? Actually, that is what is the question. Which out of these molecules, the statement is missing here, which out of these molecules will be more likely to bind to this protein? Will be more likely to be ligand for this protein? Clearly, it cannot be fatty acid because fatty acids are hydrophobic in nature. These are non-polar. And these are polar groups, positive and negative charge groups. So how can non-polar molecule interact with something which is so much charged? This cannot be the answer. Okay. Coming to cellulose, it is given in all the three options except for option 2. But to be thinking cellulose can interact with water, it can draw more of water. But you are seeing that cellulose, it's a polymer of sugar. Okay, individual glucose means it can attract water. But when it is cellulose, first of all, it's a bulky molecule, and that too, it has its properties of attracting water is decreased when it is in the form of a polymer. Okay, but DNA, RNA, if you know their backbone itself is charged, and we know that the phosphate backbone, sugar phosphate backbone that we have in DNA, the sugar phosphate backbone, the phosphate group, even after forming phosphodiester bond with the nearby sugar, still it is able to maintain a negative charge on it. Okay. So since the protein or enzyme is consists of many positive charges on the surface, it is likely that nucleic acids with a negative backbone might be the perfect ligands to bind with it. Okay, so the correct answer is option 2, which are the nucleic acids. Okay, so here is a question challenge for you. You can try to solve it by yourself. This is also regarding the techniques. All right. So a 26 residue peptide of alanine and leucine, it shows a circular dichroism spectra characteristic of alpha helix at 50 degrees C in 5 molar phosphate buffer. So they have given the CD spectra only, and you have to find out with the observations that they have given. When the peptide is gradually cooled down, the CD spectra is recorded. What will be the most likely observation? Because of these treatments, what will be the effect on the secondary structure? Basically, that's what you have to conclude. So, this is more of a protein stability question. Okay, you can let me know in the comment section. The video recording will be available later. In the comment section, you can tell me the answer. In my next series of Unit 1, we will be discussing questions on enzymology. That is another important aspect of CSIR exam. So that's all from my end. I want to remind you about the Lakshya batch. If you want to be a part of it, do contact our team today itself. If you don't get today, then you can contact tomorrow. Thank you everyone. That's all from my end.